Hey, we are Team Brainiacs. This is our capstone for the Master of Applied Data Science. Our team is made up of myself, Ben, Stacy Beck, and Mary Souls. For our project, we worked with a team at Michigan Medicine who is working on research in fMRI brain imaging and in hopes to help people who have substance abuse tendencies to better regulate the reward system in their brain. So a subject is put in the scanner, fMRI scanner, and is told to up and down regulate their reward system. And our goal is to look at these images in real time and use support vector machines and deep learning models to better understand how subjects are regulating in the scanner. Our project has mostly been split into 3 pieces. We have a single subject support vector machine where we look at individuals. We have a group model where we look at adolescents and young adults, and then we have a deep learning approach to try and get similar accuracies to the support vector machine. So our data comes in the format of 144 images while the subject is in the scanner. So as they're in the scanner, they're told to up and down regulate. So each of these images has a different label. And the subject goes into the scanner for 4 times total. In runs 1 and 4, they're not given real-time feedback, so they're not able to see how well they're regulating. And in runs 2 and 3, they can see how well they're up and down regulating the brain. So it was a pretty big pipeline to get our data all the way from straight out of the fMRI scanner into a compatible format. Mary championed the bold pre-processing. Then we needed to decide what subjects to build on. We had to remove images with no labels, mask images, so just pull out the pixels that contain the part of the brain we're interested in. Then we had to normalize and detrend, and then for each approach, there was an additional pipeline that was specific to that approach.
So for the masking, we had 3 different approaches to pulling out the voxels we were interested in. So we took a whole brain mask, which took only voxels that exist within the whole brain. Our second approach was kind of to look at regions of interest that have been kind of associated or correlated with the reward circuitry in individuals from prior research. And so we took out these regions by masking out the regions of interest. And then we also want to do the kind of the opposite approach where we trained models on just the ROIs to see if our predictions or our accuracy or our metrics change when we're just predicting on the regions of interest.
We also wanted to look at our data and make sure it would be appropriate and produce the results that we wanted when we ran it through the classifier. So we also read about trending, drifting, data drift in fMRI in time series. So the first plot there shows that the data does not have any sort of really normal distribution when it's not detrended and when it's unnormalized. So when we apply the detrending to the data, and it's still unnormalized, we can see that the variance is better, but there's still more noise in the data. We also looked at other various strategies for normalization, such as percent signal change. And when we applied that, we still noticed that the data was noisy, but when we applied a z-score normalization, which is also used in literature, we achieved a better distribution and improved the noise in the data. We can also see in this slide here how the voxel intensities appear when we apply different normalization strategies. So the top graph here the top visualization here of the brain is unnormalized, and we don't get a very good distinction between the different signals that we should be receiving for upregulation and downregulation. We get even worse outcome here with the percent signal change, where it seems all the voxel intensities are about flat. And at the bottom, when we apply the z-score normalization, you can see distinction between the up and down regulating voxel signals.
So I really worked mostly on the single subject pipeline. So SVM was performed on single subject runs data. I chose to train on run 2, which was a feedback run, and then to test on run 3, which was an additional feedback run, and then test again on run 4, which was no feedback. Before I started the process of training, I did a cross-validation and grid search on run 2 to find the best parameters for overall accuracy across subjects. The focus for single subject was to look at whole brain data to discover whether there were individual differences in model performance and to see if individuals had different activation patterns while trying to up and down regulate their brain states. We use metrics to look at both submask and ROIs to see if there were difference in predictions and how they impacted the classifiers. I also calculated metrics and created statistical visualizations to understand differences in prediction. And I created brain visualizations to kind of display the difference in voxels between classifiers. So from these two tables, this is a summary statistics looking at whole brain mass, the whole brain mass minus the regions of interest, and the ROIs that were used for model creation. I was interested in looking at run 3 versus run 4 to see if there was carryover of the training that had been done in runs 2 and 3 for maintenance of up and down regulation. And as you can see from these two tables, there seems to be a downward trend in how well the classifier is able to predict on run 4, which could suggest that there is less carryover than we'd like when we're training our individual subjects to up and down regulate. And I just also wanted to really show the spread of how accurate or how well our classifiers performed at the single subject level. We had some subjects that really performed well, and we were able to get good predictions on their increase and decrease regulation states. But we also had subjects that performed very poorly. And so this could suggest that some subjects aren't able to up and down regulate as well as we would like and may need more training. So I also want to highlight models that performed well and models that performed very poorly. So in this graph, you can see in the top panel that the high performing models were able to have really nice rock curves. And in the bottom, the poor performers were not any better than chance. So this could suggest that higher performers are better able to regulate their brain states. So this one, I wanted to look at how well the classifier decided the labels for the increase and decrease states. And so the black line is the decision function scores that were created by the classifiers. And so in the top two panels, you can see that the high performers were able, the scores more closely followed the true labels of the data in the time series. And in the bottom, it was really, there was just seemed no decision function scores that predicted. It was more flatlined. And I also want to point out that as you can see in the differences between run 3 and 4 is that we had better prediction scores on the feedback runs as opposed to the run 4. So in this one, I really kind of want to look at the Voxel distribution to see the differences in between high performers and low performers. So the top panel is the high performance classifier on an individual subject. And as you can see, there is much more differences in how the clusters are formed. We can see these bright red spots that are tightly formed clusters of voxels and clear blue spots that are tightly formed clusters of voxels that indicate increased or decreased states. And in the bottom panel, we're seeing that it's more muted and that there's not these big clusters that are distinguishing the two states.
We also ran our data by splitting the data into groups, we wanted to look at the different age groups. So the adolescents who range from 14 to 16 years old, and the young adults range from 25 to 27 years old. We're also trained on SBM classifier. We focused mainly on using the whole brain mask, but also did look into submasks and other regions of interest. But when we ran the cross-validation, we did only so on the whole brain mask data and just found the best parameters for running the models. Also, we visualized the brain using NI-Learn for the whole brain and also some other regions of interest. And we calculated and plotted the metrics and statistical tests. In these graphs here on the left-hand side, we're comparing the whole brain mask models of the young adult and the adolescents. And we can see here that the young adults are able to classify, the classifier is able to predict better. And on the right-hand side, the adolescent, we compare adolescent models and then we compare the young adult models. The young adult models are showing that each mask is predicting about the same. And the rock curve here on the adolescents, it appears that the whole brain is a little bit better at predicting than the other submask classifiers. One thing to note here is that we want to look at how each group was regulating in terms of upregulating or downregulating. And so young adults appeared to do better in terms of downregulating when expected. Adolescents are downregulating about 69% of the time when expected, and both groups are upregulating about 82%. I want to show a distinction here between the adolescent group in terms of how their decision scores are improving across runs. So in the first run here, run 2 actually, they're first receiving feedback, and you can see that subject 4 and subject 5 are separated here by the red dotted line. They're not, the decision score is not really following the true label. And so there is poor performance in terms of upregulating and downregulating. But you can see in run 3, when they receive feedback a second time and there's some carryover, you can see that they are actually carrying over as the decision scores start to improve and follow along the true label. So I just wanted to conclude here that we gathered a lot of data and we had to mainly focus on the whole brain. But we also did some statistical tests between young adult and adolescent models in terms of submask and regions of interest. And we found some interesting significance in the medial prefrontal cortex, the anterior cingulate cortex, and the anterior insula between young adult and adolescents. But we can't really come to any conclusions here. We would probably need to do more analyses using more robust statistical tests because we did not have a chance to go ahead and tune our models to the extent that we did with the whole brain.
Yeah, so we also took a deep learning approach as well, just to sort of test the waters and see if we could get similar prediction accuracies on subjects' ability to up and down regulate. It took a really similar approach to the group model, but the one consideration before stepping into building this convolutional neural network was it's a little bit hard to visualize what a subject's brain is doing in 3D space coming from a CNN, and then you need intense computing resources. But we decided to give it a go, and we built our structure very similarly to a paper written by Wang et al. And we had about 7 layers in the network and split the subjects and trained on about 36 subjects, validated on 5 and tested on 11. So in the training of the model, we had about 6 6,000 images and labels that we just individually put in and shuffled into the model. And you can see we had to split it into 8 different partitions, so 8 different training sets, just because of our limitations with RAM and computing resources. So if you look at the top right over here, the first partition and the third partition, you can see have lower accuracies, and the later partitions, the 7th and 8th training sets that we put into the model, trained very, very quickly. So that was indicative of overfitting. The model, as it ran through the images second or third time, the second or third epoch, it very quickly fit the images. So to avoid overfitting, we ran all the data through the model one more time for just a single epoch. So taking all the images and labels and pushing them through, and you can see those accuracies in the bottom right. And yeah, it looked like some of that overfitting was smoothed out. So when we put the test set through the model after training, we sadly lost 5 of the test subjects' metrics. They were deprecated, but you can see there's a pretty big span between accuracies, between 45% up to about 85%, which is actually not quite as good as the support vector machines group model. But you can see that subjects have generally similar predictions through the model. So to take another look at it, we pulled out the best and the worst predictions, similar to what Mary did in the single subject. And you can see the ROC curve of the worst prediction is about as good as random, and the best prediction is actually quite good. And on the right, the worst prediction looks just like noise, and the best prediction, the model is able to pretty well pick up the subject's ability to up and down regulate. So in concluding the CNN, we kind of just figured we need a little bit more data. So CNNs are sort of notorious for needing a lot of data to be trained, especially in 3D space with so many voxels and pixels. And it's, yeah, very easy to fall into these overfitting pitfalls. There's a lot of research out there, and deep learning is kind of newer in fMRI. And so a lot of these other studies had, tens of thousands of images that they were training the model on. But there's a lot of potential, I think, with convolutional neural networks and deep learning in fMRI space, and could be used alongside other techniques like data augmentation, transfer learning, or transformers. We also have a website. You should check it out. It's great. And we wanted to also thank all of these people who really helped us along on our project, and at Michigan Medicine and throughout the MADS program. Yeah, thank you.